This video is part of a series. It's heavy in scientific terminology and may expect familiarity with previous videos. If you're confused, please watch the rest of the playlist up until this point, or check out my genetics terminology document, both of which are linked in the description. Thanks! You'll remember in the concepts and terminology video that a complex is when a trait's expression is turned on or off by a single locus, but the appearance of that trait is affected by several modifier loci. Once again, I wish I knew the word for the monogenetic activation gene, the one that turns on the trait's expression in the phenotype, but I don't. I remember asking my most classical genetics specializing professor and not getting an answer, so personally, I call them light switch loci. The A locus is a light switch locus. It's a very simple two-allele locus with complete dominance. Capital A is dominant over little a. Capital A is Agouti, which is the yes tabby allele, the light switch is on. Little a is self, the no tabby allele, the light switch is off. The word Agouti refers to the banding of pigment on a cat's hair. If you look really closely at an individual hair from a tabby cat, possibly with the help of some kind of magnifier, you'll see alternating color bands along the shaft. With Agouti, chocolate cats become chocolate tabbies, cinnamon cats become cinnamon tabbies, so on and so forth. But black cats become brown tabbies. This is because of the aforementioned pheomelanin bands along the hair shaft that make the cat look a little bit redder. This means a tabby is not a light cat with dark stripes, but a dark cat with light between the stripes. The stripe hairs are the quote-unquote true color of the cat. Some places just appear lighter due to us seeing the banding from far away. As mentioned in the ginger video, ginger is totally epistatic to the recessive non-Agouti allele. It just ignores it. Homozygous recessive? Doesn't matter. Even a genetically self-ginger expresses the tabby phenotype and all its modifiers. Speaking of modifiers, let's talk about them. Cats with these tiger-like stripes are called mackerels. This is the mackerel pattern. It's represented by capital MC. Now, something interesting is that in genetics and development, very little separates stripes from spots or blotches. It's very easy for stripes to become spots or blotches, and vice versa. We see mutations converting one to the other all the time. Like this! You've probably seen this before. This is a king cheetah, a very cool and rare phenotype of cheetah where the spots expand and mush together into these big, beautiful blotches. Well, domestic cats have the same mutation, or an analogous mutation, meaning roughly the same change occurred in the domestic cat version of the same locus. Instead of making spots expand and mush together, it makes stripes expand and mush together. In domestic cats, this is called a blotched or a classic tabby. It's really cool that these two things result from essentially the same mutation. In scientific literature, the classic tabby / king cheetah locus is called TACPEP, but breeders just call it the MC locus, which you'll remember as being the letter used for mackerel. This is because mackerel and classic are allelic variants of the same locus. Capital MC mackerel is dominant, and little MC classic is recessive. It's recessive in cheetahs, too. I've seen some people use other letters, but these are the most common. Kinda similar to tortie patterns, but not quite the same. The exact locations and shapes of the stripes or blotches in a tabby are essentially random. The exact mechanism is too complicated for this video, but basically the pattern is produced via interactions between various chemicals. The next modifier is spotted, where the cat has little spots instead of stripes. Traditionally, breeders considered spotted to be a monogenetic trait expressed by the dominant capital SP allele that can turn both mackerel and classic phenotypes into a spotted phenotype. More recently, research has suggested that spotted is actually just mackerel being affected by polygenes, causing a kind of gradient trait that you don't really notice until it's all the way at one extreme. The theory is that mackerel itself is also a complex, and that lots of modifier loci break it up into smaller and smaller stripes until there's only spots left. To humanize, there's a kind of cutoff point where we arbitrarily decide it's more spot than stripe. While it may be an outdated model, I still sometimes like to use SP in fictional settings because human ape brain likes simple pattern, ooh-ah-ooh, but it's not strictly realistic. Here's a fun thing you can upset your friends with. Some tabbies don't have stripes. Technically, a tabby is just a cat with at least one dominant Agouti allele, regardless of their modifiers. And there's one modifier that's epistatic to every other modifier. With the presence of just one capital TA allele, mackerel, classic, and spotted are blocked. and the cat becomes... ticked! Recall that the quote-unquote true color of a cat is their stripes, and the color in between is the result of Agouti banding. Ticked cats just throw the whole keep some hairs unbanded thing out the window and go full Agouti, the whole body. Well, mostly. Ticked is incomplete dominant. Heterozygous capital TA little ta individuals will display significant striping on their legs, face, and tail. Homozygous capital TA individuals will still show the Tabby M stripes on their forehead, and sometimes stripes on their legs or tail as well. They may also have a stripe running down their back, or just a darker back in general, or they may not. You can't really see whether a cat is classic or mackerel underneath. Dominance, including codominance, only applies to interactions with alleles on the same locus. In this case, lowercase ta, not with mackerel or classic on a different locus. Being heterozygous just means that there's more stripes than usual. It doesn't affect how the stripes look. In summary, ticked is represented by capital TA and is incompletely dominant over little ta. Homozygous ticked makes a cat's hairs all but completely Agouti. Heterozygosity makes the cat a little more striped, but not by much. And homozygous recessive little ta means that ticked isn't present at all, and we can see the underlying mackerel classic or spotted pattern. I like to think of the Tabby complex like a lamp in an empty room. The MC locus is your lampshade. You have two options. The dominant mackerel, which has a stripy tiger-like pattern cut into it, or the recessive classic, which has a blotchy pattern. If you buy the mackerel lampshade, it comes with a bunch of patches of fabric that you can use to make the stripes look more spotted if you want. Or, if you want to go ticked, just don't put on the lampshade at all. No fabric pattern blocking the light, at all. Agouti is the lamp itself. Put the lampshade on the lamp, and... nothing. That's because this lamp is off. There's no Agouti allele. This is a homozygous recessive lamp. We need a dominant allele to turn it on. Great, now it's on. We can see the pattern. I chose mackerel. We can see where Agouti lights the room. We can also see dark stripes, unaffected by Agouti's lightning. Let's switch it out for classic. Pretty, look at those blotches. Now let's see what happens when we introduce capital TA to the ticked locus. The lampshade's gone. There's nothing but Agouti here. Alright, let me turn the lamp back off by making the lamp homozygous recessive on the Agouti locus again. It's all dark again, just a regular self-cat. I can do whatever I want to the modifiers. I can make the cat mackerel, or classic, or heteroticked, or homoticked, or even as spotted as I want, but you can't see it. The lamp is off. One last topic to cover here. Important to tabbies, but this also affects other cat colors. If you're here for warriors, you've probably heard the Abyssinian Firestar headcanon. Abyssinian is a breed of cat that is always homozygous ticked, but that's not all. They've also been selectively bred for unusually high Rufousing. This is a black cat. This is a black tabby. This cat is going to pass capital B black onto their offspring. There is not a single dominant O allele anywhere in this guy's genome. Ticked didn't do that. Rufousing did. Rufousing is just a fancy word for the amount of pheomelanin that a cat has, relative to what you would expect of their base color. A ginger cat doesn't have higher Rufousing than a chocolate cat inherently, but one chocolate cat might have higher Rufousing than another chocolate cat. Take a look at these two cats. One cat looks grayish and the other looks brown. Is one a gray tabby and the other is a brown tabby? No, they're both brown tabbies. If the cat was gray, aka dilute black, the stripes would be gray too. This isn't a gray cat, this is just a brown cat with really, really low Rufousing. Low amounts of pheomelanin in the bands of their Agouti hairs. The visibly brown cat has higher Rufousing, aka more pheomelanin. Can a blue cat also have high Rufousing and look reddish? Yes, actually. Here's a blue Abyssinian. They look really cool. Rufousing is a polygenetic gradient trait, so I can't give you letters for it. All I can give you is one neat trick to make your black cats look orange as hell. Doctors hate her, or however the clickbait goes. Anyway, folks who watched the multi-loci Punnett square section in the Concepts and Terminology video, your mission, should you choose to accept it, is to draw a Punnett square that includes both Agouti and at least one of its modifiers. Good luck! For the rest of you, here's your practice squares. Pause the video and figure out the chances of their kitten expressing each phenotype. 2 tabbies, 1 homozygote and 1 heterozygote. Remember, this is just the Agouti locus, so you don't have to worry about the type of tabby, just whether they are one or not. All their kittens will be tabbies. Here, we have a homozygous mackerel parent and a homozygous classic parent. Their kittens are always going to be mackerel. This is two heteroticked parents. If you said 75% ticked and 25% non-ticked, great job! But don't forget about the incomplete dominance. This pair has a 25% chance of a homoticked kitten, a 50% chance of a heteroticked kitten, and a 25% chance of a non-ticked kitten. If this is the same pair as the previous Mackerel Locus example, in which a kitten could only be Mackerel, if the kitten isn't ticked, they'll be Mackerel instead. So, this Punnett square has a 25% chance of a Mackerel kitten. If you want to learn more about blue tabbies and other dilutions, you're in luck, because the next video is on dilution. I'll see you there.